Hi, I'm SB and I'm going to explain cellular respiration. So we're going to start with glycolysis. Glycolysis is a process that occurs in the cytoplasm where we take one glucose molecule and we break it down into two pyruvates. In the process, we produce two ATPs and two NADHs. Now we'll take those two pyruvates into the matrix of the mitochondrion and we run it through the acetyl coenzyme activations. In that process, we produce two NADHs per glucose molecule, but we're going to take each pyruvate and we're going to lose one carbon in the form of carbon dioxide. And we join with a coenzyme called acetyl coenzyme A. Then we enter the Krebs cycle where we are going to take those two carbon molecules. We're going to lose the coenzyme and we're going to join with an oxaloacetate, which is a 4-carbon molecule. And we're going to make citric acid, which is a 6-carbon molecule. Now the Krebs cycle is going to eventually create again oxaloacetate. And in that process, we're going to produce 6 NADHs per glucose molecule, 2 FADHs, 2 4-carbon dioxide, and 2 ATPs. Now we create total of electron carrier 10 NADHs, 2 from the glycolysis, 2 from the acetyl coenzyme activation, and 6 from the Krebs cycle, and 2 FADH2 from the Krebs cycle. Now those electron carriers are going to release the electron into the electron transfer change, and they're going to go back down and join with half of an oxygen molecule. Now we have hydrogen ions, and they're going to join with 2 of them, the water molecule, the half a water molecule, the electrons from the electron transfer change, and they're going to create H2O, which is water. Now we started with an isotonic solution in the inner membrane space and in the matrix. But as the electron goes through the electron transfer change, they have so much energy that they're gonna pump the hydrogen ions from the matrix to the inner membrane space, which will change the concentration, making the inner membrane space a hypertonic solution and the matrix a hypotonic. Now, there are 2 major events that causes this change of concentration of hydrogen ions. The number 1 is the pumping of hydrogen ions, and number 2 is the production of H2O. Now, the hydrogen ions will want to be in a hypotonic solution, no, an isotonic solution. So they're going to go through the ATP synthase. As they do that, they will make the ATP synthase to physically spin. They will take ADP plus a phosphate and produce ATP. Now, from the electron transfer change, we're going to produce 28 ATPs. But per glucose, we're going to produce 32. 28 from the ATP, 2 from the glycolysis, and 2 from the Krebs cycle. So per glucose, we have 32.